You ready?
Do you need ours?
All right, here's a brain with the dura mater still attached, and so this thick, tough outer membrane of blue, whitish material, that's the dura mater. And you can turn it over, and some structures that are visible on the bottom with the dura mater intact are these large olfactory nerves up here at the top, and then these are the optic nerves, and they feed into a crossover point called the optic chiasm right here that's covered by the dura mater. And then this large bulge right here is the pituitary gland, and on either side of the pituitary gland are big nerves that come out, and those are the trigeminal nerves. Those are the roots of the trigeminal nerve right there. If you keep going back, there's a bulge back behind the pituitary, and that's the pons. And back behind the pons, that bulge is the medulla oblongata, and these two large ridges on either side of a midline are the pyramids of the medulla oblongata. And then back below that is the spinal cord. That tapered area behind the medulla oblongata is the spinal cord. And then if we take the dura mater off, then here's a brain with the dura mater removed. This little shiny membrane that clings to the surface of the brain is the pia mater, and what's left of the arachnoid mater has collapsed down onto the pia mater, and it appears as these dark clumps of material. That's what's left of the arachnoid mater. There's a longitudinal fissure that divides the hemispheres of the cerebrum. There's a large transverse fissure that divides the cerebrum from the cerebellum down below. Here's the cerebellum. These are the two hemispheres of the cerebellum, and then there's a central ridge of raised tissue that ties the two hemispheres together, and that's the vermis of the cerebellum right there. And then, of course, down below would be the very back end of the medulla oblongata and the spinal cord back at the back. The lobes of the cerebrum are the frontal lobe, the parietal lobe, the temporal lobes to the side, and very back at the back. You can assume these are the occipital lobes back at the back. There are some sulci that have to be identified. There is a central sulcus that runs from one side of the right hemisphere across to the left side of the hemisphere, and that divides the frontal lobes from the parietal lobes. There's also a lateral sulcus on either side that divides the parietal lobe from the temporal lobe down beneath, like that. If you take the brain and open up the transverse fissure, you can look inside and see the two superior colliculi and two inferior colliculi of the corpora quadrigemini. That is the anterior, that's really the top part of the midbrain. Then if we go to a brain that has been sagittally dissected, you can look and see, first of all, see the cerebrum, the mass of the cerebrum there. The most recognizable feature is the corpus callosum, which is a layer of white matter that forms a C shape, and beneath it is a second layer of white matter that can be separated from the corpus callosum, and that lower layer is the fornix. In between is a space that is sometimes sealed with a membrane, and the membrane is called the septum pellucidum, and my tip of my probe is going through the septum pellucidum right now, and it's going into one of the lateral ventricles. You can't see the lateral ventricles, but you can stick your probe into them as you go through the septum pellucidum. And then if you go just beneath the fornix, you will go into a space that's called the third ventricle. The third ventricle, and in the third ventricle are the blood vessels of the choroid plexus, and sometimes you can pull out those little colored blood vessels. There's one right there, and those represent the choroid plexus, and that's what generates cerebrospinal fluid. Let me switch to a different brain here. And so once again, I'm taking my pointer and I'm sticking it into the third ventricle right here. The third ventricle extends all the way back and then connects to the fourth ventricle, which is between the cerebellum and the back of the brainstem. So now my pointer's in the fourth ventricle. So this is the cerebellum right here. And the inside of the cerebellum is an area of white matter that looks like a little tree, and that's called the arbor vitae. And the gray matter that surrounds the arbor vitae is the folia of the cerebellum. Down beneath it are the pons. This enlargement of the brainstem is the pons, and back behind is the medulla oblongata, and then you can see the central canal of the spinal cord extending posteriorly from there. If I go back to the third ventricle and then go down, this egg-shaped mass where I'm moving my probe, that's the thalamus. Beneath the thalamus is a relatively undefined area, and that's the hypothalamus, but you can use the optic chiasm in front as the anterior bound and the mammillary body in back as the posterior boundary, and that tissue in between those two is the hypothalamus. There is a tiny little thread-like connection that connects the pituitary gland to the hypothalamus, and that's called the infundibulum, but in most situations you can't see the infundibulum because it's a tiny little stem of tissue that is rarely dissected perfectly. So we have to just envision that it's there. So this is the optic chiasm. That's where one of the optic nerves comes in. You can see that optic nerve there. The other one would extend toward myself. And I think that's probably all we need to cover on here. This would be this is the superior colliculus of the corpora quadrigemini. That's the inferior colliculus of the corpora quadrigemini. Let me switch to another brain section. This one has a better view of this body right here at the back of the third ventricle. That's called the pineal body or pineal gland. Present on this slice, but in that section there, it's apparently been lost. And that's it. Okay, here's the eyeball. The white outer layer is the sclera. It would in situ be covered with a real thin, clear membrane called the conjunctiva, but our model does not do a good job of showing a conjunctiva, so we just have to assume that it's there. These are the extrinsic eye muscles on the outside of the eye that move the eye around. If we look at the front here, there should be a clear window here in the front of the eye, but it's either been lost or was not there on this particular model, and so that would be the cornea. So there is no cornea on this particular model. This colored ring of tissue that surrounds the pupil, that's the iris, and of course the hole in the center of the iris is the pupil. If we take the top off of the eye, we can see the lens, which is suspended right back behind the pupil. There is a rim of elevated tissue on either side of the lens, and that's the ciliary body. The ciliary body is composed of suspensory ligaments that hold the lens in place and stretch the lens. And then there are also some ciliary processes that stick out from the ciliary body and secrete the aqueous humor that's going to fill the anterior cavity of the eye. So all of the eye that's found in front of the lens is referred to as the anterior cavity, and everything in back of the lens, back here in the back, is referred to as the posterior cavity. There is a jelly-like material in the back referred to as the vitreous gel or vitreous body, and the anterior cavity is filled with a very watery fluid, the aqueous humor. Back behind in the back of the posterior chamber, you can see this tan or orange layer, and that represents the retina. In between the retina and the sclera, there is a bluish layer, and that blue layer represents the choroid layer, which is a vascular pigmented layer. The retina, all of the axons of the retina feed into a large nerve back here at the back of the eye, and that's the optic nerve. where the optic nerve touches the retina, there is a spot in which there is no, or there are no photoreceptors, and so that generates no vision, and therefore is called the blind spot or optic disc. There's also a spot at the back of the eyeball where the photoreceptors are packed most densely, and that generates the clearest vision, and this spot back here is the center of an area called the macula. And the spot itself, where the most dense concentration of photoreceptors is referred to as the fovea centralis of the macula. I think that's everything.
That's great.
Okay, from the skull out, or actually from the what would be the eardrum or tympanic membrane out, this is referred to as the outer ear or external ear. This is the auricle or pinna, and down here's the lobe of the ear. This is the external auditory canal here, and theoretically there would be a membrane right here, and that would be the tympanic membrane or eardrum, but it has fallen off or been lost somewhere along the way. And from that point back, there's a cavity that's referred to as the middle ear or tympanic cavity, and it contains three bones, three little tiny bones, the first of which is the malleus, and that actually connects to the eardrum, the incus, which connects to the malleus, and then a third bone which is attached to this piece of the model, and this would have an extension that came out, and this is the stapes, but it has been broken off. So there theoretically, or in real life, would be an extension of this bone that came out and made it look like a little stirrup, and that's where it gets its name, stapes. Beneath the stapes is an opening referred to as the vestibule, which is part of the inner ear, and then from the vestibule are three semicircular canals, and the vestibule attaches to the cochlea, which is the organ of hearing, and then from the cochlea is a very large nerve that goes to the brain, and that's the cochlear branch of the vestibulocochlear nerve. So there's that point. There's a large tube that leaves the tympanic cavity or middle ear and goes to the throat, and that is the eustachian tube or auditory tube, can also be called the auditory tube. I think that's all of the parts that we were supposed to name.